Right, good afternoon, everyone. First of all, I would like to thank Bob and all the organizers for inviting me here. So in today's presentation, I would like to share with you our past 3 years studies on emerging swine infectious disease diagnostic and characterization. As we all know, the emerging swine infectious disease has been increased significantly during the past 2 decades. So the development of the diagnostic method and the characterization of the new emerging infectious disease and subsequently development of the vaccines and the other control strategies, they all started from the discovery of novel virus sequences. So the molecular tools, including the microarray and the sequences, contribute important roles for discovery of the emerging viral pathogens. Kevin is going to discuss about the details of the sequencing technology. So here I'm going to give you a brief overview about microarray technology that we recently adapted from the Lawrence Livermore National Laboratory. Dr. Bob Rowland was initially established collaboration with the Lawrence Livermore group. This is Dr. Crystal Jane, and she's from the Lawrence Livermore Laboratory, and she initially developed this pathogen microarray system. So the newest version of the pathogen array can detect simultaneously over 10,000 of the emerging pathogen species, and including the over 4,000 virus species and over 5,000 of bacterial species. So this microarray targets the multiple probes targeted on both conserved and unique genomic regions of the Microbial strain. So this design efficiently detecting the knowing and unknowing pathogens in a testing tube. So the microarray procedure is straightforward, and easy to be used is the automatic analysis algorithm with the web interface. So the data analysis can be initiated within 30 minutes. So this is the initial study performed by Dr. Rowland and Crystal's laboratories, and to test the feasibility to apply the pathogen array to analyze the swine samples. So they tested the serum, oral fluid, and the tonsil samples. As you can see, the oral fluid samples is the most informative sample. I would like to point out here is that the emerging swine porcine parainfluenza virus was detected in that study, which we believe it was the first detection of PPIV occurrence in the U.S. swine herd. So with the identification of novel virus sequences, the development of the diagnostic reagent and the assays are urgently needed for further diagnostic testing and the characterization of the emerging swine pathogens. So during the past years, we have developed this panel of monoclonal antibodies for different emerging swine pathogens. Some of these monoclonal antibodies have been distributed to different diagnostic and research laboratories all over the world. So with the support from the Schick grants, we have developed a quantitative RT-PCR test for this panel of the emerging swine viruses. So if you are interested in any of these PCR tests, you can discuss with the PIs for each project. And recently, we just received the Schick grants for develop the antibody detecting assays for this panel of the swine emerging viruses. So we welcome the collaboration on this project. So with the availability of the diagnostic reagent and the assays, and we were able to further isolate and characterize all these swine pathogens. So during the past 3 years, with a collaborative effort between the researchers, diagnosticians, and the field practitioners, we have characterized a panel of swine pathogens. So this including the typical porcine pastivirus, the porcine parainfluenza virus, the porcine circovirus, and the recombinant enterovirus and torovirus recombinants, and also the Silicon Valley virus. So especially we applied the basic research tools for in-depth characterization of these emerging swine pathogens. And specifically, we applied reverse genetics for in-depth characterization and facilitate the analysis of the viral RNA and the viral proteins. And this reverse genesis system also facilitates the in-depth analysis of the viral pathogenesis and host immune response. So recently, we applied our PRRS virus reverse genetics technology and to study the Silicon Valley virus. So in order to develop the full-length CDA infectious clone of the Silicon Valley virus, we used the newly emerging Silicon Valley virus strain KS15-01 that was isolated in 2015. So the full-length CDA of the SVV virus was assembled into the viral backbone, the vector backbone that we used for construct the PRRS infectious clone previously. So to further explore whether we can use this viral backbone to express foreign antigen, so we inserted the green fluorescent protein into the viral genome. So the viable recombinant viruses were recovered from the cell culture, and they showing the similar growth kinetics with the parental virus. However, when we tested in pig models, and we see initially and the similar growth characteristics from the serum sample, fecal swab, nasal swab, and oral fluid samples of the groups infected with parental virus and the cloned virus. But when we compare the clinical signs of these infected pigs, and only the group of pigs infected with the wild-type virus showing higher clinical signs, severe clinical scores in here you can see, and also the vascular lesions only showed in the group of pigs infected by the parental virus. But there was no vascular lesions was observed in the recombinant virus infected pigs. So this suggested our SVV infectious clone could be a potential useful tool to study the pathogenesis of the vascular disease in the future. On the other hand, our cloned virus showed the attenuated virulence, so that could be a potential backbone to development of the future vaccines. So using the reverse genetics, we also further explore the possibility of the molecular mechanism that underlie the emergence of the new pathogens. So I'm going to give an example in this area. Recently, we have involved in a case of the study on a naturally occurring the recombinants, which is cross-order between the viruses from the order picornavirus and the odonidalvirus. So this paper was published this year and being selected by the Journal of Biology as the spotlight feature. So we all know The recombination is a major driving force for the genetic diversity of the enterovirus. Most of the genetic recombinant events are intratypic and less happily between the virus, between the different types, and even between the different species. So the cross-order recombination is a rare event. In 2005, there was fecal samples from the pigs experiencing the diarrhea from the North Carolina sent to the Kansas State Veterinary Diagnostic Laboratories. So Dr. Bankhouse performed the next-generation sequencing analysis. Then he found a novel enter-G virus which contains about 600 nucleotide insertion at the 2C and 3A region. And interestingly, this insertion site is flanked by 2 of the 3C proteinase cleavage site. So we further isolated this virus and we performed single sequencing and we confirmed this insertion site. And then we did the plus search analysis and found out this foreign gene insertion is closely related to the torovirus PLP. So this foreign gene insertion shares about 54% to 68% with the other torovirus PLP we found in the gene bank. And the phylogenetic analysis further confirmed they formed a well-supported phylogenetic clade with other torovirus PLPs. So we designated this foreign insertion as a torovirus-derived PLP. And almost at the same time, there are 2 independent studies were reported. They found this enterovirus and torovirus recombinant from the herd at the Texas and also found from the herd at the Belgium. So the phylogenetic analysis showing this group of recombinant form a very well supported and phylogenetic clade. And interestingly, this group of the viruses are closely related to the group of the viruses found in Vietnam. So as you can see, this this group of the viruses, enter-G viruses, that were found in the Vietnam swine herds, but they do not contain the PLP gene insertion. So to further explore the function of these PLPs in the context of the recombinant virus, so we initially performed the structural analysis. And this torovirus PLP adopted a minimal core of the pipette-like proteinase fold, which is homologous to the other foot-and-mouth disease L proteinase and the SARS coronavirus PLP proteinase, and also hosts the cellular ubiquitin proteinase. This proteinase previously had been reported to possess the deubiquitination and deacetylation function, which is related in the innate immunosuppression function. So to further determine whether this torovirus PLPs has the deubiquitination and deacetylation function, we performed the in vitro cleavage assay. So the torovirus PLP protein was isolated and incubated with K63-linked polyubiquitin chain. As you can see, this torovirus-derived PLP has strong cleavage activity toward this K63 conjugated polyubiquitin chain. You can see this ubiquitin chain is cleaved into the monomer. And also this PLP has strong cleavage activities toward the ISG precursor. So to determine whether this is related to the innate immunosuppression function, and we performed the luciferase assay initially. As you can see, in cells expression the torovirus PLP and the reporter gene expression under the drive of the interferon beta promoter is suppressed. So, which is consistent showing in our RT-PCR results. So, in cells expressed the torovirus PLP, the interferon beta mRNA expression level is suppressed. So, to further determine whether this torovirus PLP's function is consistent showing in the recombinant virus under the context of viral infection, so we constructed a full-length cDNA infectious clone of this chimeric virus. And using this infectious clone, we knocked out the expression of the torovirus PLP. The viable recombinant virus were recovered from a cell culture and using our monoclonal antibodies. And as you can see, this is IFA staining results. And also the Western blot results showing the successful knockout the PLP expression from the recombinant virus. So next, this virus had a similar growth kinetics with the wild type virus. However, this PLP knockout recombinant showing the reduced growth kinetics in compared to that of the wild type virus. So we further performed the deubiquination and the deisolation assay to determine whether they still have this DUB and deisolation activity in the context of viral infection. As you can see, in those cells infected by the parental and the cloned virus, and the significant reduced level of ubiquitin conjugated host cellular proteins were observed. But in contrast, so this PLP knockout mutant is largely attenuated with this deubiquitination function. Similarly, so in cells infected by the parental and cloned virus, and the significantly reduced level of IHG15 conjugated host proteins were observed, but this PLP knockout mutant showing the impaired ability for the deisolation activity. So since the ubiquitination and isolation play important role in the signaling transduction pathway for expression of the innate immune genes, so we further tested this panel of innate immune gene expression in those recombinant virus infected cells. As you can see, in those cells infected by parental and cloned virus, and the expression of this panel of innate immune gene was suppressed. But this PLP knockout mutant had impaired ability to suppress the expression of this panel. So that means this PLP gene and function as an innate immune antagonist, and the enterovirus gene gains function through the recombinant event that enhances its ability to invade host immunity. And so these studies from these findings have the broad implication for the other picornavirus and or nidovirus species. So in summary, those studies I just presented and showing and our collaborative effort for the combating of the emerging diseases. We have adapted and our current and innovative technologies for new pathogen discovery and the development of the diagnostic assays in order to timely identify the viral infections that is key approach to prevent the spreading of new pathogens to the larger animal population. And we have also been applying the basic knowledge and research tools to understanding the viral pathogenesis and host immunity, which provides a basis to develop efficient approaches for emerging disease control. And finally, to achieve our goals, and it is essential to open interaction, communication, and collaboration among researchers and diagnosticians and practitioners. So at last, I would like to acknowledge our collaborators and our funding source.